After you've written a hypothesis and written out a prediction, then you can design your experiment. An experiment usually has several different components, and you should be able to identify these different components within an experiment. One of the major components is the presence of an independent and a dependent variable. The independent variable are values that are completely controlled and manipulated by the experimenter in order to test their effect on another variable. For example, let's just say a researcher wants to know how different types of drugs affect blood pressure. So the type of drug is what the experimenter has complete control over, so therefore the type of drug is the independent variable. Also, the independent variable tends to cause a change in another variable. So it's a type of drug, right, that is causing a change in the blood pressure. The blood pressure in this example would be the dependent variable. It's the response variable. It is dependent on the independent variable. The blood pressure is dependent on the type of drug, so the blood pressure is the dependent variable. An experiment often contains several different groups, and these groups can be divided into experimental groups and a control group. Experimental groups are groups with different levels of the independent variable, but all other factors are constant or remain the same between those different groups. So for example, if we wanted to do research on the effect of different drugs on blood pressure, the groups that receive the different types of drugs would be our experimental groups. But we would want to make sure that everything else is constant between those different groups, such as the age of the individual, the health of the individuals, the diet of those individuals, and so forth. The purpose of the experimental groups is to test the effects of only the independent variables. So that is why you only want the independent variable to be different between those groups and nothing else. It is also important to have a control group in an experiment. Now, not every experiment will have a control group, but if it's possible to have a control group, it's good to have it. The control group is going to be the group with the independent variable completely eliminated or set to some standard value. In the example of blood pressure, a group that receives no drug but instead is given a fake pill called a placebo would be a good control group. The purpose of a control group is to have a group that you can use for comparison to better understand the effect of the independent variable and also to obtain some baseline data. Let's do some practice identifying the different components of an experiment. So here we have an example. A biologist wants to know if stress during childhood changes the structure of the brain observed during young adulthood. The researchers studied 500 males. They measured the amount of childhood hardships from pregnancy to 6 years old in all the males, and compared this measurement to the amount of gray matter in the brain of each male between 18 and 21 years of age. In this case, what is the independent variable? Is there anything that the experimenters have complete control of? If there isn't anything that they have complete control over, then is there some variable that's causing a change in another variable? Well, if we look here, they're looking at how stress causes changes in the brain. And specifically what they're measuring is the amount of childhood hardships. So this is what they're measuring here, and the amount of childhood hardships is causing a change in the brain, or may cause a change in the brain. So that is our independent variable. Our dependent variable is dependent on the independent variable, and that would be the amount of gray matter in the brain. Is there a control group in this experiment? Well, in this case, there really is no control in this study. Instead, they're just surveying the natural population. What are some factors that must remain constant between the experimental groups? Some things that must remain constant, or at least must be controlled for or considered during the statistical analyses, include the diet of the individuals, their socioeconomic status, their health, and so forth. Because these are all things that actually could affect the structure of their brain. Let's look at another example. A researcher is interested in how smoking affects a person's risk of developing lung cancer. They monitor how often lung cancer appears in 3 different groups of people over a 20-year period. The first group are non-smokers, the second group are light smokers, and the third group are heavy smokers. So what is the independent variable here? Well, the independent variable here, so what is causing a change in something else? Well, that would be the amount the individuals smoke. The dependent variable, so what is dependent on the amount they smoke? Well, it's the prevalence of lung cancer, whether or not they get lung cancer. The control group, do we have a control in this case? Well, we do have a group where the independent variable is eliminated, and that is the group of non-smokers. So that's our control group. So we want to know, well, how often does lung cancer appear in non-smokers? And to see if it really is greater in individuals that smoke. What are some factors that must remain constant between the experimental groups? So again, we have to make sure the only thing really that's different between these different groups is the amount they smoke. Everything else should be constant. So that includes the diet of these individuals, their socioeconomic status, their health, age, whether they're male, female, their physical activity, and so forth. After you design your experiment and you collect all this data, and then you have to analyze your data. When scientists analyze data, they typically use statistical analyses to analyze that data. And I'm not going to get into details about statistical tests in this video. However, I do want to talk about the importance of statistical analyses. It is extremely important to have some basic understanding of statistics because this allows scientists to make sense of and summarize large amounts of data. It also allows them to see if a set of data supports their hypothesis or not. Furthermore, it allows scientists to determine the relationship between different phenomena. Is there a correlation between two different variables? And statistical analyses allow scientists to predict future trends. After a scientist analyzes their data, then they can use these statistical tests to determine or make a conclusion, and this conclusion states whether their data supports their hypothesis or not. Then the final step of the scientific process is publishing. And publishing can be one of the most tedious steps of the whole scientific process because after you've collected all your data, run all your experiments, then you have to write everything up. So now hopefully you have a better understanding of the scientific process and what scientists do every day.